my friend Dr. Kellis, and he has graced us with his presence today. I like to usually do the introductions, but if let's see, I'm going to say this and you tell me if it's correct because uh, you are a computer scientist and computer biologist known for your work in the field of computational biology and genomics, right?
Yep,
And do you still hold a position of professor at MIT, or is that
correct.
okay? You do. Okay, excellent. So, Dr. Kellis's research primarily focuses on developing computational methods and algorithms to analyze and interpret like genomic data, the human genome. He has made significant contributions to understanding the organization and function of the human genome, as well as the genomes of other organisms. So, his work includes the development of many different innovative algorithms for identifying functional elements in the genome, understanding gene regulation, deciphering the genetic basis of various diseases, etc. So, this is what we're going to talk about tonight. I have a set of questions, of course, for you, my friend. But I usually like to ask people, what are you most into right now? What are you most interested in? What is the project that has your eye?
Well, we have four papers coming out in the journal Cell on Thursday, and this is pretty exciting. It's about the dissection of Alzheimer's disease at single cell resolution, basically trying to understand what is the molecular basis of Alzheimer's, what are all of the different signatures, what are the cell types that are implicated, what are the pathways that are altered. And this is sort of one of the areas that I'm just very, very excited about, basically trying to understand what is the circuitry, the molecular basis of human disease. And by understanding human disease, can we figure out where to intervene? What are the pathways that we should alter? What are the genes that we should be targeting? What are the regulators? And that's where computer science comes in, basically by understanding the circuits that underlie these genetic associations. We are then able to recognize targets, recognize pathways, recognize
Mm-hmm.
how we should be altering. And those four papers are basically telling us that there's these dramatic alterations that are happening that are underlying resilience, for example. So a lot of individuals that have symptoms of AD have all of the molecular signatures, all of the pathological signatures, and all of the cognitive signatures. And this is the traditional definition of Alzheimer's. But there are other individuals that show all of the pathology when they're dead. You basically look at their brain and you're like, oh my God, half the brain was gone. They have this enormous pathology. They have these amyloid plaques and these neurofibrillary tangles. But cognitively, nobody knew that they were suffering from Alzheimer's. They didn't show the cognitive decline. So
Yeah,
one of the
behaviors weren't there.
papers is basically telling us about resilience, about the ability to sort of withstand these dramatic changes to your brain without actually showing any phenotypes.
Yeah.
Another one of the papers is basically telling us how the immune cells of the brain, known as microglia, how they are rewired during Alzheimer's disease, how they become inflammatory, and what are the circuits, what are the regulators that we can use to turn them back to homeostatic state, so turn them away from inflammation, which is very damaging. The third paper is basically telling us about epigenome erosion. So many of the cells that are found in Alzheimer's individuals, they lose their identity. They are showing this flattening of the epigenome. So as you know, the DNA in all of our cells, we have trillions of cells, and the DNA in all of them is the same.
Yeah.
But every single one of our cells has very different functions. So what we are studying is how this is possible, how microglia and neurons and excitatory neurons versus inhibitory neurons,
Yes.
oligodendrocytes, astrocytes, the
Yes.
vascular cells, how all of these cells are able to, with a single common genome, show such dramatically different functions. And then what we're showing is that some of these processes are in fact altered during Alzheimer's disease, possibly related to DNA damage, and that the epigenome is in fact eroding, and sort of all of these memory of the cellular identity is going away. And that basically leads to this de-identification of these cells and then ultimately the loss of their functionality. So anyway,
Does
these
that
are sort of
include
the different
neurons?
aspects.
Oh, exactly. Does that include neurons dying and not being
Yeah,
able to be put
absolutely.
back in?
So
Not being,
basically,
yeah,
the most cells in our body are constantly renewed.
yes.
There's this great metaphor of a
7
ship,
years.
where if you replace every plank of
Yeah,
that ship, is it
yeah.
still the same ship at the end?
Ship of Theseus. Yeah,
And exactly. And then with the same metaphor, you can think of the human body as constantly renewing itself. Basically, every one of our cells will eventually be renewed.
yeah.
The one exception, the cells that you do not want to renew, are your brain cells, because that's where the memories are stored. So basically, if you sort of renew your neurons, you will no longer be you. You will no longer have your memories. So the ability to sort of turn over neurons is one of the things that unfortunately is not possible. So what we need is constant repair rather than renewal, rather than
Is
replacement.
there a future in optogenetics in that way?
So what optogenetics allows us to do is effectively reprogram individual cells using light stimuli.
Right.
And it's extremely helpful for model organisms. It's not something that we are envisioning for humans to be able to sort of, go in and sort of
Yeah.
create these switches, if you wish. But what that allows you to do is have programmable individual neurons that you can kind of turn on and turn off at will. And
Yeah.
that's usually used in mice or in model organisms that allows you to sort of study these circuits by intervention. But there are other ways of intervening on the brain, which is using, for example, electrical stimuli, or using optical stimuli, or auditory stimuli, or magnetic stimuli. So basically, there's a lot of startups that are basically looking at different ways of intervening in addition to the traditional chemical ways, just because of the electrical properties of the brain.
Yeah. No, that's awesome. Did you mention the fourth one, or did I mishear you?
Yeah, so basically
I may have
the
interrupted
third
you. Go
the third paper was about epigenome erosion, and the fourth paper is about DNA damage. So what's very interesting and sometimes surprising, possibly to many of your viewers, is that part of the way in which memories are encoded in the brain is by actually making cuts on the DNA itself. So basically, the DNA double-stranded breaks is actually part of the process of memory formation. And most of the time, they are repaired accurately. But with the progression of Alzheimer's, sometimes those breaks are no longer repaired correctly.
Okay.
And what we're showing is that sometimes the increase in DNA damage basically leads to genes being fused together from different parts of the genome. And that can then lead to altered gene functions, sometimes altered gene regulation, because the regulatory control elements of one gene will be together with another gene.
Yeah.
So basically, the fourth paper is looking at DNA damage and sort of how that is altering
What other besides Alzheimer's
Alzheimer's.
disease? What can DNA damage do to us?
So DNA damage is involved in cancer, for example, but
Hmm.
there it's a programmed beneficial change that is actually beneficiary to the cancer cells.
Oh,
The idea there is that most of our cells are very careful about their genome. They want to sort of make sure that there's no changes, make sure that it is very, you know, accurately
mhm.
copied from
Yeah.
one cell to the next. With cancer, however, what is beneficiary to cancer is that you can have massive dysregulation, because that might actually turn on genes that are helping the cancer replicate more, or that are helping the cancer gain new functions. For example, evading the immune system, or sort of turning on angiogenesis to basically sort of bring more energy to
Yeah.
itself, etc. So in the case of cancer, this DNA damage is actually self-reinforcing. So basically, the more mutations you have, the more likely you are to be replicating. For neurons, it's a little different, because the neurons don't replicate. So basically, simply having neurons, replicate is not something that sort of is self-reinforcing. So
Yeah.
the DNA damage there is sort of more of a passive event, and simply possibly the result of this accumulation of DNA damage, and at some point, possibly coupled with the epigenome erosion, sort of being able to sort of going past the point of no return in terms of the accumulation of that damage, and
Okay.
and
So
there it
yeah.
kind of leads to this very detrimental end stage processes. But most of the time, neurons will not simply apoptose, like self-suicide, the
Yeah.
way that most other cells will, just because we kind of like keeping on to our memories.
Yeah, you keep the. So could you explain, just I guess in your way, because it'll be much more professional than anybody else could explain it, what neurogenesis is to the audience?
So the way that our brain forms is truly extraordinary. So most of our body forms through a series of sort of replication event and specialization of the different cells. So every person starts from a sperm and an egg meeting and then forming the zygote. And then the zygote undergoes a series of cell divisions, sort of forming in mass, of course,
Hmm.
always receiving the nutrient from the mother
Right.
or from the egg in sort of precursor species before mammals. So dinosaurs and lizards, etc. They lay eggs instead. But
Right.
basically, the egg for mammals is inside the mother. So basically, all of the nutrients are constantly supplanted there. And what's really extraordinary about the human brain. So basically, we know a lot about development of different body plans. So basically, very initially, there's this body plan formation with segmentation. Basically, different segments are associated with, for example, the head, and then the dorsal to ventral
Yes.
axis and anterior posterior axis. So there's two axes of development, and they're sort of defining different segments of the body. And there's these homeobox genes, these Hox gene clusters that define the different segments, and they're kind of laid out in order in the genome, and they're also laid out in order in the patterning of the body, and that sort of gives the gross or organization of the body plan. But then for the brain itself, it is quite extraordinary because we basically have trillions of neuronal connections, and we simply don't have enough genes to program them all. This is not like in C. elegans in the nematode, in the worm. We basically have exactly a predetermined set of cell divisions that give rise to exactly the number of 300 some neurons that the male and the female worms have. In human, it's very different. We basically have neuronal connections forming in projections that are not necessarily individually programmed, but instead project to different regions. And there's
Okay.
morphogens and all kinds of stimuli that sort of lead to that. And in addition, during the differentiation process of the neurons, you basically have neuronal migration. So the way
Oh.
that you form all of the different regions of the brain is that you have formation of the neurons and then migration sometimes along the vasculature. And for a long time we've thought that new neurons do not form, but more and more we're realizing that there's continued neurogenesis even in adulthood sometimes. With, for example, the hippocampus shows formation of
Yes.
actually new neurons, and I think we still have a lot to learn about sort of when does neurogenesis end,
Oh, yeah.
and when does the sort of stable brain
Yeah.
finalize. But
Is there
then there's
neurogenesis going
constantly
on in a dying human? You know?
uh, probably not neurogenesis,
Yeah.
but throughout our life, we basically have synaptic pruning. So basically, these are the synaptic connections between the different neurons that are constantly getting cut if they're not being utilized, and then reinforced if they're being utilized. So this whole
Right.
concept of use it or lose it for a lot of our habits and memory formation, etc., is very much associated with this whole ability to rewire these connections. And that there's a lot of parallels of that with the way that we train neural networks and artificial neural
Mhm.
networks in deep learning,
Yeah.
where these neuronal connections, these weight updates, there's algorithms that allow you to do that. And this is enabling the artificial neurons to learn patterns that they observe based on reward of connections that are bringing you closer to the ultimate goal, and then penalization of connections that push you away from it. And in the
Yeah.
same way, humans basically update their neuronal connections, not necessarily through neurogenesis, but through reinforcement of specific
Right.
connections,
The
which are
connections
then,
form more and more. Yeah.
yeah, so these connections are basically learning specific concepts, learning specific, input output relationships, and sort of forming ideas, thoughts, concepts, memories,
Yeah.
every aspect of human cognition. is instantiated through these primitive processes.
Yeah. So it's somewhat akin to a biological imperative. It sounds like. um, so it's interesting because you bring up Alzheimer's. We can talk about that more because it seems as though your papers are focused on that. Um, what happens to like the neural connections, the neural pathway connections on someone like my grandmother who is 90 and has Alzheimer's?
So
That may have not been the best question, but I think you
no,
get.
no, no. So basically, the way to understand Alzheimer's is the same way that you should understand, early life growth, embryonic development, childhood, adolescence, and adulthood. Basically, our brain is a combination of software and hardware. It's a combination of experiences and the underlying hardware that encodes sort of these connections. And the analogy that I like to give is that we kind of live in two different time scales. So if I throw a ball at you, you will catch it, immediately. Even from the corner of your eye, if you perceive it, you'll immediately catch it. This happens in, millisecond time scales.
Right.
The way that we think, the way that we respond, the way that we speak, if you watch table tennis, for example, or gymnastics, etc., there's some programmed behaviors, and sort of there's sort of a motor learning, component, and the cerebellum in our brain is sort of
Right. Yeah.
involved in a lot of
Fine
this motor
movement.
learning.
Mm-hmm.
And then in addition, there's this sort of logic and inference and reasoning. And the our habits, for example, are associated with specific neuronal pathways that are being formed, reinforced, rewarded, and strengthened. And this is happening at timescales that are extremely slow.
Yeah.
By contrast, when I throw a ball at you and you catch it, this happens at timescales that are extremely fast. So physiologically, life undergoes transcription, RNA expression, epigenome activation in the order of minutes, sometimes hours. And you can think of that as the hardware changes that are kind of implementing the underlying circuitry upon which all of these electrical signals and stimuli, which are much, much faster, will be happening.
Yeah.
And, if you look across different kingdoms of life, you will see that, for example, plants, if you accelerate them by a factor of 100, then you can kind of see them moving, reacting, looking at sunlight, turning, etc.
Right.
So life has like all of these different time scales at which it operates. Now, if you look at the changes that are happening during the adolescence, you basically see changes that are, on one hand, happening in the learning itself at the electrical signal, which is then translated to chemical signals of synaptic connections being reinforced, strengthened, etc. And then that ultimately translates to actual hardware changes in the actual circuitry of your brain. So the and sort of that's why it's extremely important that children are exposed to enriching experiences, early in their childhood, because then they're kind of forming these neural connections that will then, help them in late life.
Yes.
And now to look at Alzheimer's, for example, if you have an enriching childhood, that actually gives you increased resilience, increased reserve against cognitive decline. So basically, the early life experiences that your grandmother had are actually playing out now in late life in terms of cognitive reserve.
Okay.
And the way to understand the late stage of life is that we have this extraordinary machine between our ears that's basically running 100 times faster than many of our relatives. In evolutionary timescales, this happened extremely fast. And there's metabolic adaptations that humans had to kind of undergo in order to achieve that. And if you look at the resilience of a brain that undergoes injury, it basically switches from mitochondrial energy production to directly burning of sugar, glycolysis, and instead of oxidative phosphorylation. And OXPHOS basically requires sort of an energy grid, if you wish. And neurons that undergo injury are instead switching to sort of, burning the stove in the countryside, so a much sort of less efficient form of energy. So injury in the brain will basically accelerate ultimately neurodegeneration late in life and will lead to tau accumulation, neurofibrillary tangles, and sort of these signatures of damage that are also associated with the end result of amyloid accumulation, which is associated with neuroinflammation, with inflammatory processing, your microglia, and so on and so forth. So basically, if you're looking at sort of these early life, late life changes in the hardware and this rewiring of the underlying physical instantiation of our circuitry, of our neuronal circuitry, you can kind of start interpreting now those events of cognitive decline, of cognitive resilience, and so on and so forth in the context of those hardware changes. I hope that answers your question.
No, it does, and you can map it out, right? It's a way to at least get some kind of a map uh, to understanding. Here's a crazy question: Could we ever CRISPR away Alzheimer's?
So let's talk about CRISPR for a second. So
Yeah.
what is CRISPR? CRISPR is a way to cut out very specifically specific regions of the DNA. I didn't say edit, I said cut, because the technology itself is co-opted from a bacterial immune system against viruses. So when bacteria are infected by viruses, these these bacteria kind of store a signature of the virus, so that next time that virus comes, they're able to go and cut the DNA or the RNA of that virus. So CRISPR was originally discovered in bacteria when they were sort of trying to make yogurt,
Yeah.
grow more efficiently. And what CRISPR does is that it has this programmable array that allows you to now cut very specifically based on a sequence signature of the virus to cut that virus.
Gotcha.
We have, as engineers, co-opted this to sort of allow replacement of these viral signatures to basically be any location in the genome that you'd like to cut. And what that leads to then is a pathway of DNA repair, where you have a double-stranded break where you can kind of repair it. And the way that genome editing is usually implemented is by combining this CRISPR ability to cut very specifically a piece of DNA with band-aids that we throw that basically have a complementary sequence to the one you're cutting with the specific letter that you'd like to replace replaced.
Yeah.
Now, when you start thinking about sort of CRISPRing out diseases, the way that you think about this is that there are strong effect genetic variants that will sort of predispose someone to a disease, and that you will somehow be able to edit those variants. For most diseases, the variants are, the genetic differences are playing a very, very small role. There are some exceptions. So for example, for obesity, there's a single locus that adds 7 pounds to your adult weight if you're homozygous for that locus. This is an enormous effect size. Most of the variants have tiny little effect sizes. But this one is an exception. FTO in obesity is an exception. ApoE for Alzheimer's is also an exception. So ApoE4, the E4 allele of the apolipoprotein E, ApoE gene, the E4 allele for ApoE4 individuals increases your risk by a factor of 10. So if you are a carrier of 2 copies, one from mom, one from dad, of the ApoE4 allele, then you have a 10-fold increased risk. Wow. So the actor that plays Thor in the Avengers, Chris Hemsworth, was recently sort of came out November last year, basically saying, I am homozygous risk for ApoE4. Actually, on the same day that our paper studying the effects of ApoE4 appeared in the journal Nature. And what we showed is that individuals who carry the E4 allele have altered gene expression in very specific pathways in multiple cell types. And the one that we focused on that paper was basically showing that oligodendrocytes which are the cells that protect the axons of neurons
Yes.
that coat them with myelin,
Yeah, the
this
sheath.
sort of,
Yeah. Yeah.
exactly, the myelin sheath. So what we showed is that the transport of myelin, the transport of cholesterol, which is one of the ingredients that is required to sort of form myelin and to protect the neurons, that transport was impaired. And we also showed that you can kind of add these hydrophobic molecules that allow you to facilitate transport, and that when you restore transport, transport of cholesterol, you restore myelination, and you restore cognition in animal models. So
Hmm.
this actually provides enormous hope that perhaps we can kind of restore a broken process. And the way to understand now this in the context of a late stage disease like Alzheimer's
Yeah.
is to think about what initially causes the disease, and then what happens after your brain has undergone so much damage. So this is a disease that is late onset. Individuals have Alzheimer's by 50, 60, 70, 80 years old.
Yeah.
So that's decades of damage before the symptoms even appear.
Hmm.
So by the time somebody's diagnosed with dementia, with cognitive decline, they have been losing neurons for decades.
Yeah.
So the question is that a good time to go and CRISPR, quote unquote, out the
Right.
disease?
When would be the demarcation line? Yeah.
That's exactly right. So basically, at what point can you intervene and still restore normal physiological function?
What if you cut out something important too?
Well, that's true for any disease.
Yeah.
Basically, for any therapeutic, not just for CRISPR. Basically, very often we're kind of interested about off-target effects and
Yeah.
sort of
That's what happens with me. I'm bipolar, and all the medication I take is off-target effects. It's a proxy. Affect this to affect this.
yeah, yeah.
The neurochemicals and everything. But no, yeah, I'm with you. Um, well, we can keep talking about this because I don't understand a lot about. I understand. I would say I understand 70% of the terms here, so I'm just being generous with
Yeah,
that.
yeah. No, feel
um,
free to ask away
yeah, no,
because I'm
I
sure
love
that
you.
your
I love
listeners will
you
be
both.
wondering about the same terms.
Yeah, if you want to explain, go deep into anything you can. We just love learning here, and you can speak as high level as you want to. We love it. So,
Yeah.
um, uh, so is there a way? Is like the myelin sheath? Is that like a super? That's super important, right? Can we repair that at all right now?
So there was a paper recently about multiple sclerosis. So this is a case where in the peripheral nervous system you basically have this loss of myelination. Similarly, and that's when the myelin is actually getting attacked by your T cells. So it's an autoimmune disorder when the myelin is sort of targeted as an enemy and sort of getting digested away. So the the corresponding effect is basically a lot of myelin loss in Alzheimer's, and of course the myelin is extremely important in being able to protect the electrical signals,
Yeah.
because without it the signals themselves will be losing and attenuating their strength, and therefore the signaling will not be as clean, and you will get sort of crosstalk between different neurons and between different synapses and leakage of these signals.
Oh. Okay.
That's one aspect. So basically, this is the physiological aspect.
Yeah.
The other aspect is that the myelin also protects neurons from injury. So in the
Mm.
case of Alzheimer's disease, in the case of neurodegeneration, having this myelin sheath actually protects the neurons from damage. So there's a double whammy there. On one hand, the signals themselves will not sort of travel as easily. On the other hand, the actual circuitry of your neuronal connections is actually falling apart.
Mm. Is there any medicine to affect this?
So you can aim for two things. Number one, sort of stop the killing, and number two, restore the dead. So basically, stop the myelin from being lost, and then number two, restore the myelination itself.
Yeah.
And there are both existing medicines and sort of therapeutics in development that try to address both sides of that equation.
Oh,
I feel like we lost Thomas. I can see some of the comments on the chat. It says protein buildup preventing nervous system communication. We have to look at the difference of binary logic versus biological knowledge, logic. That's very cool. Okay, so you're saying that the experience of engine is causing the physiological problems for the engine for the future. RNA replication, splicing. Yep, he's reloading. Um, so yeah, I want to go back a little bit to the concept of CRISPR and being able to edit individual genetic variants. So the way that I think about this is that we're not going to be editing diseases away by eliminating individual variants, because many of these variants for complex traits are in fact very weak effect variants. For rare diseases where you know that you almost have a sort of destiny, where the effect sizes are very large, then that makes a lot of sense. And there's several companies that are sort of focused on doing that, very rightfully so. Um, but for the vast majority of complex traits, these effects are very small. So the way that I think about these therapeutic development is not through individual variant edits, but instead we use the genetic variants to establish causality, to establish the circuitry. And then once we have established that, then we're able to go and understand how are these variants acting? What cell types do they act in? What are the genes that they affect? What are the upstream regulators? What are the pathways and biological processes? And when we understand all that, then we're able to um, target those in a sort of in a way that has a much stronger effect. So I'm wondering if I should join the new link there. Okay, so I'll just stay on. Okay, no problem. Yeah, no worry. So I don't know if you have any questions. I'm happy to dive into those. So he asked me earlier what things I'm excited about. So I mentioned the 4 papers that are coming out in Cell, and they're all about Alzheimer's. We have another paper that's coming out in Nature Genetics, also on the same day. And then that paper is looking at genetic variation in brain, heart, muscle, and lung, and how that is associated with changes in the epigenome across hundreds of individuals. So this is part of the GTEx project, the Genotype Tissue Expression project, and that's allowing us to understand how are genetic variants ultimately manifesting into disease impact, and what are the intermediate molecular changes that are associated with those. Hey, Thomas, are you back?
hey, hey, Dr. Kellis. Yeah, my internet is terrible, man. I don't
No, it's
understand
okay.
what's going on. No, yeah, it sounded like y'all were talking about something interesting. If you want to expound upon that, please go ahead.
No, basically, you asked me what am I very excited about, and I mentioned the 4 papers, but there's a lot of things that I'm very excited about. One of the
Well,
things that
keep
I'm
going.
excited about is basically now that we have many of these targets for understanding the causality of these different disorders, understanding the way in which they impact gene regulation, the way they impact the cellular circuitry of the cell, we now have the ability to say, okay, great. Now that we have this target, how do we develop a therapeutic for it? And traditionally, this has taken decades, basically from the initial discovery of the target to sort of designing the drug, optimizing the drug, etc. And this is a place where we are trying to shorten that from 10 years to a few months. And the way that we're doing this is leveraging the power of large language models, of protein language models, of graph neural networks and geometric learning,
Mm.
and this extraordinary power of generative AI. And in particular, what we're doing is we are training models that have as input, on one hand, the folding of a protein, on the other hand, the description, the biological description of the function of that protein. And in a project that we're carrying out in collaboration with Marinka Zitnik at the Harvard Medical School and Brad Penteluta at Harvard at the MIT Chemistry Department, what we're basically doing is, on one hand, using the genomics and the genetics and the epigenomics to understand the circuitry and understand the target genes and the pathways and the biological processes. On the other hand, designing new folds using this joint learning, this contrastive learning, in the same way that you can kind of learn how images and text go together by finding pairs of image and text that are the same and
Mm.
pairs of image and text that are discrepant, and sort of training them based on the difference between these close pairs and these far pairs, known as contrastive learning.
Yeah.
What we're doing there is learning effectively the protein folding language of protein function. Namely, we are trying to understand what aspects of the structure are in fact responsible for the function of different proteins. What aspects of these structures are in fact matching drugs that are interacting with these proteins.
Yeah.
And using the two together to be able to reason in the English language about chemicals that can inhibit those particular folds, about what are the folds that are associated with normal physiological function or with disease functions, and so on and so forth. So basically, what I'm very excited about is sort of bringing the power of these generative models to the service of medicine,
Mm-hmm.
to be able to now start speeding up that process
Yes.
where we find the target, we design the therapeutic, and then in collaboration with Brad and his team, where we can very rapidly synthesize both the chemicals and the protein domains that we predict they should be bound to in the order of hours instead of months,
But you need that
to be able to
algorithm
sort
for
of
the
then
folding, right?
test.
The folding. You
Correct.
need. Yeah. Okay.
So you've heard about AlphaFold and the extraordinary achievements that machine learning has made to be able to sort of fold proteins, to effectively learn the language of protein folding, something that humans over the course of decades and thousands of hours of experts studying these sequences have not been able to do. But now that AI is able to do so, it's kind of very exciting to sort of be living at a time where so many different aspects of the circuitry of our brains and of our bodies and of our cells is in fact getting cracked open,
Yeah.
and it's kind of very, very exciting
It is
to
exciting. I
be
mean,
at that place.
see, I mean, I've just recently, I'm a philosophy psychology nerd, and so I've just recently learned about genomics because I talked to Christopher Mason. Have you ever heard him?
Yeah, he's awesome.
Yeah, he is awesome. He was great to talk to. That's where I learned about epigenetics and genomics and things like that. Um, did you fall into this? What originally made you want to do this?
So, as an undergraduate, I studied computer science. So, I'm very much fascinated with AI and machine learning, and that's what my undergrad and my master's were on.
Mm.
It's the first year of my grad school that I kind of switched to biology, and it was through evolution. So I always wondered about genetic algorithms
Yeah.
and how, like this extraordinary elegant model of Darwin that sort of basically couples random mutation with brutal selection. So, the mutation, the generation of diversity is purely random, mutations occurring throughout the genome. And then the adaptation to different environments happens through simply survival of the fittest.
Right.
And this is very elegant when you're kind of building computer programs that are trying to sort of find solutions by
Yeah. Like
combining parts.
evolutionary algorithms, that kind of thing.
Correct, genetic
Yeah. Yeah.
algorithms.
Yeah.
And basically, what bothered me is that if you look at the timescales of evolutionary adaptations, it basically took us 1 billion years for single cell organisms to, figure out all the basic machinery.
Hmm.
And then the acceleration towards multicellularity, complex body plans, all of these evolution of organs and the specialization of all of these different hundreds of cell types in the human body
Yeah.
and in the human brain, that happened in the blink of an eye
Yeah.
evolutionarily.
In deep time. Yeah.
So
Absolutely.
with genetic algorithms, this should not be possible because as you increase complexity, the space of solutions becomes so large that finding the beneficiary ones is very difficult.
Yeah.
So, my first foray into genomics was basically studying evolution and looking for number one, evolvability. Basically, maybe the first 1 billion years we kind of got good at evolving. We kind of discovered modular structures for genome organization, making sure that the space of solutions is not mostly leading you to non-functional things, but that there's a language for DNA that then allows most mutations to effectively be
Hmm.
uh, tolerable and to sort of have that robustness, that modularity that then allows for evolvability. So that's sort of, how I got into this theoretically. And then my first papers in the field were all about understanding evolutionary signatures, understanding the patterns of change between different genomes to both understand how genomes evolve and to also be able to read evolution's notebook in order to discover functional elements in the genome. So I discovered a
Yeah.
series of evolutionary signatures, lenses, if you wish, that allow you to discover where are the protein coding genes, where are the non-coding regulatory motifs, where are the RNAs, the microRNAs, etc., in the genome, and using those to effectively, for the first time, understand the human genome de novo. When I was an undergrad, we didn't even have the human genome. Basically, in
No. Yeah.
'99 is when the
Yeah.
whole complete sequence was annotated.
That's right. Yeah.
And, the challenge ahead of
Francis
us was how do we
Collins, right?
understand that?
Yeah.
Yeah, so Francis Collins was the head of the National Human Genome Research Institute, NHGRI, at the time before he became the head of the NIH.
Yeah.
And then Eric Lander and many others on the public side, and then Craig Venter on the private side. So, it's 20 years ago that we basically finally had the sequence of the human genome. And
It's
so much has happened since then. It's just an
exactly.
extraordinary revolution.
Yeah, and quantum computing is on the horizon. I know we have to cool it down to like ridiculous temperatures, and IBM has one the size of a car. But I mean, isn't that made theoretically for like computing genetic algorithms because there's so many variables?
Yeah, there's some similarities there, but I would say the two fields are quite disconnected. So,
Okay. Yeah, I mean, I don't know anything about quantum. I know about quantum computing, but not about computers. I don't know what they require. So I didn't know if you were even. It's probably years from you even using any tools like that to for, to learn more about the genome or learn more about the genetic algorithms that you study. I imagine,
I would
right?
say that they're mostly of theoretical interest right now rather
Yeah,
than of practical interest,
yeah,
and many applications in cryptography, but not so much in genomics.
Okay. So, well, like with genomics and stuff, you're just using classical computing, just throwing a heckload of computing power at this, right?
Yeah, so we're using CPUs, but also GPUs for a lot of the deep learning
Yeah.
applications.
Yeah.
So
That's really cool. That's cool. That I mean, I love looking at the brain, and this is a very low resolution take for me compared to what you've been saying. But I always look at the brain as hardware and like the mind as software. You know, just as like a heuristic, even though it may not be exactly like that. Also, some people automatically think that because DNA is a code, that means it's intending to transmit a language from an agency to another agency, and it's like no, DNA is analogously a code, right? Or a language.
well, there's many transformations and many codes at many layers inside our body. So at the base, you do have DNA that codes for protein, for example.
Hmm.
And there, it is truly a code. There's a genetic code that allows you to translate the nucleotide sequence into an amino acid sequence.
Okay.
And, you can think of it as assembly code that you're then sort of translating into um, a program that you run that eventually folds into 3 dimensions and makes a protein.
Yeah.
That's a base layer. And then on top of that, there's the epigenome. So the epigenome is kind of interpreting the DNA and turning on and turning off different parts, poising the other parts, sort of marking repressed regions and heterochromatic regions and transcribed
That's what
regions
the
and
epigenome is
promoters and
doing.
enhancers. That's what the epigenome is doing. It's basically marking the regions that will be important for every
Okay.
cell type to maintain its function.
Right.
And then there's a code there as well. Then there's the code of how different parts of the DNA evolve over time, how different types of functional elements evolve over time. There's a code for the positioning of the nucleosomes. There's a code for the epigenomic modifications that are happening both at the histone proteins, but also the epitranscriptomic modifications that are happening at the RNA.
Yeah. Describe that word.
And let me just add a few more of those.
Go ahead.
So
Go ahead.
basically, all of that is the base layer. And then there's the whole code of development of basically what are the patterning processes and the signals and the receptors and the logic for creating the body plan.
Hmm.
And then there's a whole other layer which is encoded in our brains for storing information, for experiential learning, etc. So it's truly beautiful to sort of think about all of these different layers coexisting. And as we're trying to understand, for example, the impact of Alzheimer's on cognition, how much of that is due to the neuronal encoding of information versus the epigenomic, DNA breaks and genome rearrangements and genomic and epigenomic organization within every one of these neurons versus the DNA code or the nucleosome positioning code and all of the regulatory code, all the developmental code. So it's it's
Yeah.
it's just very beautiful to sort of think about all of these different
No, I see
layers
what you
of encoding
mean.
coexisting,
Yeah. It's
each
it's all
living in
it's
its own time scale.
in its own time scale. Yeah. Exactly. Do you remember that word that you said? Could you describe that to me? Because I don't remember it now that you said it. Oh man.
It was
Because
about nucleosome positioning and epigenomics and
there was
histone
like a
modifications.
That's okay. I won't remember it because you said it was really specific, and I thought I would ask. I didn't mean to interrupt you in your flow there, but
No, no problem.
no, I see how it can be seen as a code, and I get that there are different, many different levels that you just described very elegantly about that code. I was just making a pithy comment about how sometimes people will talk about DNA as being
Thomas, are you still there? It's okay. So I kind of have to go as well. So I think I'll take the opportunity to thank you guys for the chance to speak with you tonight, and I look forward to seeing many more of your episodes.
Yes, we will share the episode with you. Thank you very much,
Awesome.
Dr.
Thank
Kellis.
you.
Yeah, thank you.
Good night. Bye.
Good night.